And I thought Emma Griffith did a great job in communicating that the intent for GMI is a focus on the why. Right, welcome everyone to another episode of the Bioinformatics Lab podcast. I'm Kevin Libuit with Andrew Page, and today we're going to talk GMI 14. Andrew, you and I were both at GMI 13. Do you remember much of it? What your experience is walking out of
Yeah.
GMI 13, that beautiful city in Vancouver?
Well, GMI 13, that was Global Microbial Identifier. Vancouver is fantastic, but there was only about 50, 60 people there in a lovely little venue, but it's all the right people, and that's what matters, bringing together the right people internationally to talk about how we share data and what do we need to do and what do we need to change. And you got to go this time to Barcelona, so tell me all about that.
Yeah, that's right. And I like the way GMI has been doing that. This is the second time I've gone to the conference, you and I going out there to Vancouver for GMI 13. And Barcelona kept that same spirit of a small, really concentrated, really forward-leaning cohort of attendees from all over the world. It's kind of mad how a small venue like that, again, maybe 50, 60 people representing the forefront of the field, from WHO and IPSN with Josefina Campos, FDA being represented by a number of folks from Eric Steven, Mark Allard, Ruth Timmy, and then a whole host of just the who's who, really, of the people pushing things forward on both the implementation side, the workforce development side, and even the technical side. There was, obviously a lot of bioinformatics scientists there as well, including Torsten Seaman, right? So like the whole Torstiverse was represented there, and it kept the same spirit of GMI 13 in terms of just addressing one, the progress made throughout the field, looking at the major challenges, and also pretty forward looking as well throughout some of the talks. For example, there was one session I think you would have really dug. It was a focus on AI and implementation. Yeah, yeah. It was interesting. It was cool to see, and all the different ways in which people are trying to push that technology in its applications in microbial bioinformatics. And oh man, I'm actually kind of curious to get your take on this. There was a hotly debated item a bit on the importance of metadata. And on one side, as you can imagine, from I would say represented by or led by Emma Griffith and Ruth Timmy and all the folks on the data standards group at Phage, of the importance of metadata and all the fantastic arguments they've made with respect to being able to track outbreaks, understand the geographic time, and all the context really around the samples that are being made available. Torsten took the hot take,
Yeah, I
and
mean,
he, yeah, go on, please.
metadata has been a hot topic for a long time, and
Yeah,
particularly ontologies for actually describing things, because you can have all the AI in the world, but you need the fundamental data to train it all on. And obviously, bacteria don't know where they are located, and you can have so many different curveballs in there. Like, say, a sample is collected from a traveler who went to Africa and then comes back to the UK, and it can be often recorded as the UK when actually it's incorrect, and then source attribution becomes this huge car crash basically.
and that was actually a big point too of what a lot of the AI conversations were, because the richness of metadata, but a lot of it is just um, it's just people saying the data. It's unconfirmed. It's based on, it's almost like survey data. It's like, okay, this is the patient information, and we're just going to trust you. So the models, therefore, downstream are just trusting the metadata. But I take a step back there. Torsten took a hot take. It was awesome. He said, metadata is not that important. All I need is maybe a country and a point of contact. And that's the most important information. And from there, at least I know who to call to conduct my investigation. And I would say standing on that side was more so Mark Allard and Torsten, their major argument being that, one, if I have a point of contact, I can conduct my investigation. And two, it lowers the burden of data sharing. It's like, just get the data out there, a point of contact, and you don't necessarily, which is often the case, need to coordinate all these different efforts to make sure you have the appropriate metadata in play prior to submitting to these international databases. So yeah, putting that in your lap. I'm curious what your take is, if you can kind of weigh out those differing opinions.
I guess one issue is that if you don't capture the metadata or upload the metadata with the sequence at that time, then it's never going to get updated at all. And, you'll be there for 10 years later, trying to back populate it. And I know when I was in the Sanger Institute, we did have one person dedicated to basically taking all the supplementary material spreadsheets that we'd publish and then try and back populate them onto the actual biosamples years later, because originally the samples were created before even sequencing, that you just create this kind of shell, and then you go and do sequencing, you publish the paper and the sequence is right there, but you wouldn't publish the metadata or take that backwards step. So it was a huge amount of effort to go backwards. And people move on, people forget things, people can't remember why they called something something, and you lose all of that. And like, I guess back in the day, it was somewhat easier with no privacy restrictions and whatnot, where people would just name the sample after the person who died from that particular thing, and you could go back to medical records. Yeah. One of my colleagues went back, and it's the very first sample deposited in the National Collection of Tissue Cultures in the UK, so like the ATCC, except for the UK. And there was a guy called Cable. It was just, the sample was called Cable because it's after Private Cable, who died from dysentery, you know? And so they could actually go back to the archives, the military archives, and then pull out this guy's records, and then you'll find out where he served and lived and died, and which hospitals he's in, and who collected the samples. And, huge amount of rich information, like 100 years later, could be pulled out because they had that link, and they're able to go back and back populate basically the metadata. But now privacy restrictions are obviously such that you have an obscure label, and you can't do that backward step, and it can get lost along the way. But it's an interesting take on that, particularly since people don't want to share data. They might just jump at that and say, oh, well, good excuse.
I'm glad he said it. I love that we had the forum for that kind of a disagreement just to take place, because I've been hearing the metadata, share the metadata, and I buy in rather, to the sharing of the metadata. But it's interesting to see that position put out. So it kind of forces me in a position of like, ooh, okay, wait a minute, why do I disagree with that? And I hear the conversation of pragmatism, just get the data up there completely. But I'm just of the camp of there's so many more downstream applications when that rich metadata is made available beyond just a point of contact. And exactly to your point too, if it's if you're able to share with sharing limited metadata, like the other data is never going to make it there. And maybe the fear too or apprehension is that data being maintained otherwise as well. Even if you're making that point of contact, is that information that will be accessible. That's kind of what you're banking on, is if you're able to make that phone call, that there's there's metadata there to be shared otherwise. But then even getting back into the context of the conversation of AI and what could be learned and intuited when you have that strong metadata, we lose all that if all you have is a point of contact there. So I love that Torsten and Mark Allard actually, I think, shared that same perspective, which was really fantastic, strong. I'm glad that they voiced it. Though on my end, I'm still very much an advocate of sharing the metadata. There's such a richness to the genomic epidemiology that could be made available when all that information is shared, and distributed and made available to the community. But beyond that, GMI, there was also big emphasis on the role they play in the community, against juxtaposed to maybe the phage organizations, the spheres, and all the like. And I thought Emma Griffith did a great job in communicating that the intent for GMI is a focus on the why, in advocating and being able to make resources that highlight why we're doing this. And I thought that was a great point. And she mentioned that this is the type of content or resources that promote sustainability. So when people are speaking to policymakers and decision makers and funders, they can point to resources that maybe GMI helps to curate that tell them why. Why is this important? What are the economic arguments? Brad from, I forget his last name, from the FDA, presented on his assessment of PulseNet program in the US and the return of investment, and that being an obvious why as to what this saves lives and money. That's an easy why to get behind. The case studies in terms of solved outbreaks, and all the different resources and ways in which people are able to implement these technologies. So I love that as the spirit of GMI, is a focus on the why and curating resources, getting people connected so we can emphasize why we're doing this.
So will you be going back to GMI 15 in Melbourne next year?
Oh man! At the end, just similar to in Vancouver, at the end, the next host made a spiel and sort of an advertisement of the next conference, and this was something done by Tia and Torsten from MDU. They made a good argument. Like Melbourne looks like a great place to be a part of, so definitely on high on the list of next year being able to attend GMI 15. So,
Awesome. I'll see you there then.
all right. So with that, I will see everyone in the next one.